Applying Mendel's principles. What is the chance your children will all have brown eyes? Whenever Mendel performed a cross with a pea plant, he carefully categorized and counted the offspring. For example, whenever he crossed two plants that were hybrid for a stem height, tall and short, capital T, small t, about 3/4 of the resulting plants were tall and about 1/4 were short. 3/4, 1/4. Mendel realized that the principles of probability could be used to explain the results of his genetic crosses. Probability is the likelihood that a particular event will occur. For example, there are two possible outcomes of a coin flip. The coin may land either heads up or tails up. The chance or the probability of either outcome is equal. Therefore, the probability that a single coin flip will land heads up is 1 chance in 2. This amounts to 1/2 or 50%. If you flip a coin 3 times in a row, what is the probability that it will land heads up every time? Each coin flip is an independent event with 1 chance in 2 probability of landing heads up. Therefore, the probability of flipping 3 heads in a row is 1/2 times 1/2 times 1/2, and that would be 1/8 or 12.5% chance of having 3 heads in a row. As you see, you have 1 chance in 8 of flipping heads 3 times in a row. Past outcomes do not affect future ones. So the chance of a heads on that fourth, even if you've already had 3 heads, just because you have flipped 3 heads in a row does not mean that you're more likely to have a coin land tails up in the next flip. Every single flip is 50-50, no matter what the history. The way in which two different alleles segregate during gamete formation has been seen to be consistently the same as a random coin flip, 50-50. Therefore, the principles of probability can be used to predict the outcomes of genetic crosses. If each F1 plant has 1 tall allele and 1 short allele, capital T, small t, then half of the gametes they produce would carry the short allele, small t. So capital T, small t, half is here, half is here. Mendel's cross produced a mixture of tall and short plants. Mixture of tall and short. Because the small t, the short allele, is recessive, the only way to produce a short small t plant is for 2 gametes carrying the small t allele to combine. Small t and small t. Each F2 gamete has a 1 in 2 or 1/2 chance of carrying the small t allele. There are 2 gametes, so the probability of each gamete carrying the small t allele is 1/2 times 1/2, which is 1/4 or 25%. Roughly 1/4 of the F2 offspring should be short, and the remaining 3/4 should be tall. This predicted ratio, 3 dominant to 1 recessive, shows up consistently in Mendel's experiments. For each of the 7 crosses, about 3/4 of the plants showed the trait controlled by the dominant allele. About 1/4 of the plants showed the trait controlled by the recessive allele. Not all organisms with the same characteristics have the same combination of alleles. In the F1 cross, both the capital T and capital T small t, the tall and the tall short allele combinations resulted in tall T plants. These three, all three are tall T plants. The small t, the short allele combination produced a short P plant. Each organism has a genetic makeup as well as set of observable characteristics. All of the tall P plants, capital T, tall, and a tall short allele combinations, had the same phenotype or physical traits. However, all of the tall P plants did not have the same genotype or genetic makeup. One had capital T, and the other capital T small t, the tall allele and the tall short alleles. There are three different genotypes among the F2 plants: tall short, tall, and short short. The genotype of an organism is inherited, whereas the phenotype is formed as a result of both the environment and the genotype. If they are stunted by poor nutrition, they may look like a small t, short, but actually they're tall t, or capital small. Two organisms may have the same phenotype, but different genotypes. Organisms that have two identical alleles for a particular gene, tall, or short, in this example are homozygous. Homozygous is both capital T's, also homozygous is both small t's. Organisms that have two different alleles for the same gene, such as capital T, small t, are heterozygous. These two in the middle are heterozygous. Probabilities predict the average outcome of a large number of events. The larger number of offspring, the closer the result will be to the predicted values. If an F2 generation contains just 3 or 4 offspring, it may not match Mendel's ratios. When the F2 generation contains hundreds or thousands of individuals, the ratios usually come very close to matching Mendel's predictions. One of the best ways to predict the outcome of a genetic cross is by drawing a simple diagram known as a Punnett square. Here's a Punnett square. Punnett squares allow you to predict the genotype and phenotype combinations in genetic crosses using mathematical probabilities. So how to make a Punnett square? Determine what alleles would be found in all the possible gametes that each parent could produce. Write the genotypes of the two organisms that will serve as parents in a cross. In this example, we will cross a male and a female osprey that are heterozygous for large beaks. They each have genotypes capital B, small b. So here's our source, parent, and a parent. Large beak, small beak, large beak, small beak. So because these are dominant, both parents have large beaks. So we'll draw a table with enough space for each of the paired gametes for each parent. Each of the genotypes of the gametes produced by both parents on the top and left side of the table. So here's one parent. Here's a gamete here. So here's an allele for one parent, and allele for the other parent. Fill in the table by combining the gametes' genotypes. So filling in the box, we take each of these in the column and fill in here, each of the boxes, and a small b would fill in each of the rows. So completing the box would be capital, and this capital would be the capitals here, and the smalls would be here, and the small would be here. In order to determine the genotype and phenotype of each offspring, calculate the percentage of each. In this example, 3/4 of the chicks will have large beaks because of the dominant capital B in these three boxes. One of two will be heterozygous, either small b, capital B, or capital B, small b, and the order does not matter. Mendel wondered if the segregation of one pair of alleles affects another pair of alleles. Does one characteristic affect the other? Mendel performed an experiment that followed two different genes as they passed from one generation to the next. Because it involved two different genes, Mendel's experiment is known as two-factor cross or dihybrid cross. Single gene crosses are monohybrid crosses. Mendel crossed two breeding plants that produced only round yellow peas with plants that produced wrinkled green peas. Wrinkled green peas crossed with round yellow peas and produced the F1 generation. And because round yellow are dominant, all F1 generation were the dominant characteristics, both round and yellow. The round yellow peas had the genotype RR, capital R, capital Y, which is homozygous dominant. The wrinkled green peas had the genotype small r, small y, which is homozygous recessive. All of the F1 offspring produce round yellow peas. These results show that the alleles for yellow and round peas are dominant over the alleles for green and wrinkled peas. The Punnett square shows that the genotype of each F1 offspring was capital R, capital Y, small y, heterozygous for both seed shape and seed color for all the F1 generation. Mendel then crossed the F1 plants to produce F2 offspring, F1 and F1. And these are the allele combinations from the F1, every possible option. Same here. So F2 had a larger mix of options as the alleles combined. Mendel observed that 315 of the F2 generation were round and yellow, while another 32 seeds were wrinkled and green, the two parental phenotypes. So these were yellow, and only 32 were wrinkled and green. But 209 seeds had combinations of the phenotypes, and therefore combinations of the alleles that were not found in either parent. The alleles for seed shape segregated independently for the seed color alleles. Genes that segregated independently, such as the genes for seed shape and seed color in pea plants, do not influence each other's inheritance. So Mendel's experiment results were very close to a 9:3:3:1 ratio that the Punnett square predicts. Mendel had discovered the principle of independent assortment. The principle of independent assortment states that the genes for different traits can segregate independently during gamete formation. So 9 round and yellow, one's in round and yellow, but then there were 3 round and green, and there were 3 wrinkled and yellow, these 3, and then finally 1 wrinkled and green, a ratio of 9, 3, 3, and 1. What did Mendel contribute to our understanding of genetics? Mendel's principles of heredity, observed through patterns of inheritance, form a basis of modern genetics. The inheritance of biological characteristics is determined by individual units called genes, which are passed from parent to offspring. Where 2 or more alleles, or the forms of a gene for a single trait exist, some forms of the gene may be dominant and others may be recessive. In most sexually reproducing organisms, each adult has 2 copies of each gene, one from each parent. These genes segregate from each other when gametes are formed. Gametes are formed, they separate each characteristic. Alleles for different genes usually segregate independently of each other. At the beginning of the 1900s, American geneticist Thomas Hunt Morgan decided to use the common fruit fly as a model organism in his genetics experiments. The fruit fly was an ideal organism for genetics because it could produce plenty of offspring, and it did so quickly in the laboratory. Before long, Morgan and other biologists had tested every one of Mendel's principles and learned that they applied not just to pea plants, but to other organisms as well. The basic principles of Mendelian genetics can be used to study the inheritance of human traits and to calculate the probability of certain traits appearing in the next generation.